PCR in forensic sciences. While discussing the different modification of the basic PCR reaction, like the RFLP or AFLP, we discussed that these variation or applications of the PCR, they can be used in the field of forensic sciences. So forensic sciences, it is the application of the scientific procedures in order to relate a particular individuals to the crime scene. So scientific methods they are used, and in case of molecular biology, the information that is generated from the human genome sequence, it can also be used to relate a specific person with some specific crime scene. And this technique, now we can also call it as the DNA profiling, or more common term that is used, it is DNA fingerprinting. As I told you earlier, that initially fingerprints were used to relate certain individuals with certain legal matters. But here, with the advancement in the molecular techniques or approaches, now the information from the genomic material, it can also be used. And because of this reason, a term which is called as DNA profiling or DNA fingerprinting, it has been used. मिसाल के तौर पे क्राइम सीन से डिफरेंट इंफॉर्मेशन हासिल हो सकती है, like blood samples, saliva sample, tissue sample, or some other samples. They can also be collected either at the crime scene or from the victims. And now we want to relate that a particular victim, it is responsible for the situation or crime. Now, how we can do that? We can do that by using DNA fingerprinting in forensic sciences. DNA fingerprinting, I just explained you that here we can use the restriction fragment length polymorphism and its modified form AFLP we can use. So we can extract the genomic DNA from the evidence or tissue or blood sample that we have collected, and then we can digest it with restriction enzyme after the extraction of genomic DNA. So genomic DNA is there, we have a particular restriction enzyme, we can digest the DNA that have been extracted from different sources. Like DNA, it can be extracted from the tissue that is collected from the crime scene, from the suspect, or from the victim, or different suspects that are involved. As I told you that due to polymorphism of the DNA, that DNA of human, it can show variation up to 0.1%, and this 0.1% variation, it can influence the target sites of the restriction enzymes. Like in one individual, the restriction enzyme pattern or the target sites, they may be located differently as compared to other human individual. So these variation patterns, it can be exploited. So when we will try to digest the DNA from one individual and compare it with an other individual, it will show slight variations in the bending pattern on the gel, so that we can relate specific person with specific legal matters. And after separation on the gel, I told you that other procedures, they can also be used. So combinations of procedure we have to apply, like extraction process must be there, then separation on the gel, then it's transfer to the nylon membrane, then autoradiography we have to perform to detect the DNA fragment. So although this technique, it may be a bit time consuming or laborious, but effective in order to get the profiling of the human genome. So this is one of the application in the forensic sciences. In addition to that, some other legal methods that one can encounter in daily life, like in order to establish the paternity, one can also use PCR or DNA fingerprinting technique. And here again, the paternity can be established by extracting the DNA from the child, from parents, and in this way we can say that this particular child, it belongs to a particular parent. So paternity matters can also be solved by using this technique of PCR.